all to engage and participate and cheer, so do not be shy. Because part of what our celebration today is to celebrate Rex's 80th birthday. So my buying time while we are getting Zoom set up is we are going to all enthusiastically, loudly, but quickly sing happy birthday to Rex. So ready?
Yes.
Happy birthday to you. Happy birthday to you. Happy birthday, dear Rex. Happy birthday to you. Excellent. So I don't want to take up too much time, but I do want you all to keep your energy up. So if you are post-lunch needing to have a nap, the only rule is that you have to channel Rex, and it has to be a 5-minute power nap, and you need to come out of it. We're ready to roll. So those are the guidelines. Otherwise, we are going to start off our first session here with a plenary by Jane Marks. Fabulous presenter. I need not say anything more. I'm going to hand over the mic to you. You can take it over.
Thank you. This is hard. I can barely use one hand, yet let alone two. Okay, I can do it. April 12th, 2024 was a really important day for me. I was at the Stanford campus visiting some colleagues, and when I walked into Ellen Yee's lab, she handed me the recent copy of Science, where they described the discovery of a nitrogen-fixing organelle. And I was actually at Stanford that Friday because my daughter Nona is a student there, and she was directing the student production of SpongeBob SquarePants, the musical. And being the good theater mom that I am, I sat through 4 shows. But all the while, during intermissions, I'm on my phone reading through the science article, which was the proto-organelles biochemistry. But after 4 shows, I felt that I gleaned the main points. SpongeBob and his friends needed to save Bikini Bottom from an emerging volcano. And also, we have a new organelle. That doesn't happen every day. About 100 million years ago, a marine haptophyte, a eukaryotic algae, engulfed the cyanobacteria, and eventually most of the genes of the cyanobacteria are now on the nucleus of the haptophyte, encoded in its cytoplasm, and then transported into a new organelle, the gold standard of being an organelle. And while this is a huge step for humankind, it was personally meaningful to me because I, like many other people here, study another alga, Epithemia, that also has a nitrogen-fixing or endosymbiont. I'm going to be careful with my words. The cyanobacterial endosymbiont has a reduced genome. It's lost both photosystems 1 and 2, so it cannot photosynthesize, but it does have the genes to fix nitrogen. It is vertically transmitted, and to the best of our knowledge, we never find the epithemias, any of the species that are in the Robolodia family without the symbiont, and we never find the symbiont without the diatom. My colleagues at Stanford invited me into this paper that was also published this year, where they coined a new phrase, diazoplast, to differentiate the cyanobacteria, which is a very close relative to the one that's in the organelle, but we're differentiated from the nitroplast because it doesn't have quite the level of genetic integration. But where it lies between the continuum of 2 organisms, one living inside another, and one organism is up for debate. But nevertheless, it's now emerging as a model system for understanding organellogenesis, which is a word. A lot of the work's being done by molecular biologists and biochemists. Some of the work that's being done by people in this room is focusing on the systematics and taxonomy of the Rhopalodias. But I'm interested in how it functions in nature. How does Epithemia and the symbiont, does it give it an advantage over the hundreds of other photosynthesizing algae and the hundreds of other nitrogen-fixing prokaryotes that it lives with in a river ecosystem? Does it have an outsized role in carbon and nitrogen cycling and in structuring the food web? Just like SpongeBob lives in a pineapple under the sea, in the ecosystem I'm working in, the Eel River, Epithemia lives on Cladophora in the Rio. This is Mary Power. I'm sure many of you are familiar with her work. And what she and others, including Paula and Rex, have shown is that Cladophora dominates the primary producers in this. There's many, many rivers worldwide. But it goes through a very predictable succession of epiphytes. In the spring, it looks green to the eye. It doesn't have many epiphytes. Then it becomes lightly epiphytes, primarily by Coccinius and Mycosphenia. And then eventually it's smothered by Epithemia. And it goes through what I think it was Paula who coined the stoplight. It goes from green to yellow to red. So I'm going to use that metaphor throughout this talk as it gets more and more epiphytized. Under the microscope, green Cladophora can grow to be meters long. It's highly branched, has a thick cellulose cell wall that becomes epiphytized. You can see Coccinius. Let me see if I can coordinate this. Coccinius and Mycosphenia. And then ultimately, I stole this slide from Rex, happy birthday to me, that shows in this shot that it's just covered with epithemia cells with the cyanobacteria diazoplast inside it. So my tripartite symbiosis is going to focus on Cladophora, Epithemia, and the diazoplast, but in the context of the complex food web in which it lives. Even though the phenology is unidirectional, you can find all the stages at different places in the river at the same time, depending on light levels and temperature levels. So we can collect algae, bring it back into the lab, and then keep everything else constant other than the stage of the epiphyte assemblage. This is CE, he's somewhere here, he better read, there he is. This is some of a slice of his dissertation work. We're using 18S first to look at the epiphyte assemblage. So don't waste any neurons in trying to differentiate these three pie charts from each other because they're the same. Whenever we collect the Cladophora at this coarse level, it's about between 2/3 and 3/4 diatoms, a little bit of other algae, and then generally a 3-to-1 ratio of autotrophs to heterotrophs. Focusing on the diatoms, we see it's largely a Coccinius epithemia story. So it starts with Coccinius dominating the diatom assemblage, and then more and more epiphytes take over, both in terms of absolute and relative abundance. I'm going to focus now on carbon and nitrogen fixation, two big elements in biogeochemistry, and report a series of experiments, all of which use stable isotopes. So we label the algae with C13CO2 and N15N2 to focus in on carbon fixation or photosynthesis and nitrogen fixation. And what we find over and over again is this pattern, that as Cladophora becomes more and more epiphytized, the rate of photosynthesis per gram of algae goes down. Concurrently, the rate of nitrogen fixation goes up. So why? Why is carbon fixation going down, and who's fixing all this nitrogen? So to dig a little deeper, we turn to nanoSIMS, which brings the power of stable isotope tracing with the high resolution of scanning electron microscopy. So we can look at carbon and nitrogen fixation on a cellular or intracellular scale. Let me show you an example. What we do is we take Cladophora and we cut a cross section, put it into the nanoSIMS machine, which generates an electron micrograph and a heat map of carbon C13 and N15. So the more red to yellow you are, the more C13 you have, and similarly with N15. So let's start with green algae, or the green Cladophora, and we're going to go around, clockwise around. So we take a cross section, and this is what the micrograph looks like. You can see the cell wall, and you can see the reticulate chloroplast. When we look at the carbon heat map, you see the carbon fixation is almost entirely concentrated in the reticulate chloroplast, and then you can also see the blue in the cell wall. So it's investing a decent amount of recently fixed carbon into maintaining its thick cellulose cell wall. Nitrogen, lights out. There's no N fixation going on or getting into the Cladophora. Let's move in time a little bit to a slice of yellow, and I'm picked here a representative slice that has two Epithemia diatoms. So this is the Cladophora. It looks like kind of a distorted bowling pin. And here are the two epithemia cells. And with the carbon, what we see is that Cladophora is still fixing at a slightly lower rate, but so are the epithemias. But when we go to the N15, we start seeing some action. So this is recent fixed nitrogen concentrated in the two epithemia diatoms. Again, little to none is getting through into the Cladophora. At the red stage, we see here this halo of diatoms all around. And then you can see that it's hard because the lighting's not so great here, but there's very, very little C13 in the Cladophora cells. So it is dying. It is inactive. It's not photosynthesizing anymore. All the photosynthesis is in the epiphytes. And similarly, all of the nitrogen fixation is in that halo of epiphytes. We can also look at it at a cellular level and trace the carbon that's being fixed by the diatom. Remember, the diazoplast, the symbiont, cannot fix carbon. So we can trace it from the diatom to the symbiont. And similarly, we can trace the fixed nitrogen from the symbiont or the diazoplast into the cytoplasm. So here's, I'll show two representative cells. Here's the first one. This is the cell, and you can see the imprint of the symbiont. And there it is, getting a ton of carbon from the diatom. And then you can see it fixing a lot of nitrogen and sharing, in this case, a little bit into the cytoplasm. So it's blue, not black. So some of it's going into the cytoplasm. The next one shows a more generous symbiont. You can see the symbiont here, and again, getting the carbon and then sharing more of its nitrogen through the diatom. So we've got multiple cells of these that show a very tight coupling of carbon fixation and nitrogen fixation and sharing of the carbon with the symbiont and nitrogen with the diatom, presumably giving it the big competitive advantage where it gets to be occupying up to 90% of the diatom assemblage. Okay, so that explains in part why carbon fixation is going down, because it was per gram of algae. And if half the algae in that circle is Cladophora, it's not photosynthesizing. So that explains that. So essentially, Cladophora's role in the ecosystem shifts midsummer from being the main primary producer to being, it essentially goes detrital on us, but it has the decency to decompose slowly providing tremendous surface area for the diatom, which gives us, I'm not showing aerial rates, but the rates of infixation in the Yo River are really very, very high because Cladophora provides all of this surface area that takes a good long time to decompose or export out of the system. But what about this high nitrogen fixation? There's a lot more there's a lot more prokaryotes in a wad of Cladophora. This is Michael Wolf, who's also a student in my lab and here today has helped with all of this research. There's thousands and thousands of prokaryotes, right? So the diazoplasts are a small piece of the prokaryotic community. And we know now there's over 200 that can fix nitrogen. This is a close-up of a Epithemia frustral, and you can just see it's loaded with diatoms. I'm going to do close-up just because it's so adorable, of which about 20% of them have the capacity to fix nitrogen. So we set out to partition nitrogen fixation among the free-living bacteria and archaea, almost entirely bacteria in this system, the free-living cyanobacteria, and the diazoplasts. And so to do this, we assumed that the contribution to nitrogen fixation is directly proportional to both the biomass of each individual and fixer and the rate at which it's replicating or growing. So we can get biomass through 16S sequencing, but figuring out how quickly microbes are growing in situ, because this is what we care about, what's it doing in nature, not what it's doing in culture by itself, is extremely difficult. So members of the group that I work with at Northern Arizona University have developed new protocols called quantitative stable isotope probing. We nickname it QSIP, that measures species-specific growth rates of microbes in situ. And I've tried to explain it every which way. And so I finally resorted to creating this crazy video inspired by Rex, because it's all about candy. That's 2 minutes, so I'm going to show it here. Let's go. A new tool called quantitative stable isotope probing, which differentiates growing from dormant microbes. So this is a big advance in microbial ecology. How does it work? Well, it turns out that all organisms, including microbes, when they replicate, take in oxygen from water. So if we grow microbes in water that's enriched with 18O, we can trace the heavy isotope into the microbes that are growing. But the difference in weight isn't very big. You can't measure it on a vacuum scale or on any scale for that matter. You need to do it through a centrifuge in cesium chloride. If you grow microbes in normal water, they have different weights based on their nucleotide composition. Let me demonstrate. If we had a microbial community with just 4 taxa, we would start with our blue peanut M&Ms. We would also find a closely related sister taxa, the orange peanut M&Ms. We might find yellow plain M&Ms, which are related, but a little more distinct, but still part of the M&M family. And then we could also find distantly related good and plenties. If we were to take these taxa and put them in a centrifuge and spin it, we might end up with something like this. They would distribute based on their weights. The blue ones weigh the most, the orange ones weigh the least. But if we were to grow the same community in 18O, we might find this. So let's unpack it. First thing you see is that the orange taxa didn't move at all. They're not growing. Neither did the blue taxa. The yellow moved a little bit, so they're growing slowly. But look at these pink. They grew good and plenty. They grew a lot of the 18O isotope, meaning they're growing a lot. But to get this data, we need to sequence 15 layers in this tube and 15 layers in this tube. Okay, so that's QSIP in a nutshell. So I just showed is if we had a community of 4 taxa, what we really have is a community of thousands of taxa. And so I'm going to pull out from Raina's work just the nitrogen fixers. So we did the QSIP for our free-living archaean bacteria, our free-living cyanobacteria, and then also the diazoplasts, because they have a 16S signal, just like any of the prokaryotes. And diazoplasts grow they can grow both by creating more diazoplasts within an epithemia cell and also when the epithemia cells divide. And I'm lumping them together in this talk. Okay, so we predicted that the number of nitrogen fixers would go up, and it did. It's about 50 taxa in the green Cladophora, lightly epiphytized Cladophora, and pops up to over 200 in red Cladophora. We expected to see more 16S of nitrogen fixers, and we did, and we expected to see more cells of nitrogen fixers. So let's go there. So we see an increase in nitrogen fixers as we go from green to red, and nitrogen fixation goes up. But we also see that about 3/4 of them are free-living bacteria and archaea. But when we translate this to biomass, the pattern shifts a little, and we see that earlier on, it's still mostly free-living or cyanobacteria, but by the time we get to the highly epiphytized, it's about 2/3 biomass are in the diazoplasts. When we look at the growth rates, these are just the means, but I have growth rates for every single one of the bacteria species. The growth rates are about 3 times higher for the free-living bacteria and archaea than the free-living cyanobacteria or the diazoplasts, which makes sense because they're much smaller cells. So when we put all this into our model to partition nitrogen fixation, this is what we find, is that in the early stages, N fixation is about 2/3 done by the diazoplast, but by the time we get to the red stage, when it's really cranking, it's about 90%. Of the nitrogen fixation going on in the river at that time is due to the diazoplast. So that's a lot. It's a lot of assumptions, some of the problems that we talked about with DNA work, plus it's a lot of multiplication, so that if I'm wrong, I'm going to be exponentially wrong in a hurry. And so I'm smiling on the outside, but worried on the inside. So I did a kind of back of the envelope check to see if we're in the ballpark. And I reasoned that N fixation in red Cladophora, highly epiphytized, is about 6 times higher than in yellow Cladophora. And so that if I'm right, and that the increase in N fixation is largely driven by this increase in diazoplasts, then we would expect also a 6-fold increase in diazoplasts between red and yellow Cladophora. So I did that fairly simple math and came up with in the ballpark. And I'm also now in a very good mood because one of my graduate students who's not here, Michael Ciampini, has shown that epithemia functions as a superfood in the Eel River. And what Mike did was he took our labeled algae and fed it, different kinds of labeled algae, and fed it to a grazing caddisfly gumaga. And so he did the 3, the green, the yellow, the red, and then he expanded it to very, very late season where sometimes it gets covered by Anabina, and then we added nostoc and Spirogyra into the mix. There's gumaga. This is a grazing caddisfly. It's everywhere, and it does well in our laboratory mesocosm. So we trace the isotope into the gumaga. Okay, carbon. You've seen this. Photosynthesis or carbon fixation goes down from green to yellow to the red. So let's put it in context to the other algae. What we find is that the cladophoras are lower than either the nostoc or the spirogyra. So they're photosynthesizing at a higher rate. But what are the insects assimilating? Red Cladophora, Epithemia. So much, much higher than any of the other types. Nostoc wasn't a surprise because only that little midge eats nostoc. But so much more carbon is going into these insects from the epithemia than from anything else. When I standardize it to efficiency, the percent of carbon that's being fixed that goes up, it's hovering at 1% for these, like 2.5% here, but it's like at a whopping 10%. Like that is a really efficient system. 10% of the carbon that's being fixed is going up the food chain if it's packaged in epithemia. Nitrogen stories similar. Here are the nitrogen fixation rates, so no surprises. Highest here, the red Cladophora, but then the Anabina adds to the nitrogen fixation. Nostoc's kind of right in the ballpark, and not surprising, there's very few nitrogen fixing bacteria embedded in the spirogyra. And then we do the same thing, and we see a lot more nitrogen is being transferred up the food chain when it's Cladophora covered with epithemia. So what did I learn? Well, SpongeBob and his two best friends in a tripartite symbiosis saved Bikini Bottom. And I set out to ask, does the symbiosis confer a competitive advantage and have an outsized role in the ecosystem? I'm going to go with yes. At least for like a month or two during the summer, which is like the salmon rearing time in this river, and Epithemia symbiont is really driving carbon and nitrogen influx into the river, and carbon and nitrate fluxes up into the macroscopic food chain. I can end it there. I've got this is 3 quick slides of where we're going next. This is all work in progress. This is Augustine, who couldn't come today. But some of the work we're doing is we set up this whole facility to manipulate temperature and nutrients. And so we're now that we've got all kind of the kinks worked out, we're looking at how temperature, nitrogen, and phosphorus affect all the dynamics that I just showed you about. We're also doing some scaling experiments to look at the Cladophora and the epithemia in the context of everything else that's going in the river, and seeing, like, when we measure things in little bottles, do they scale up to the reach scale kinds of measurements, where we see a lot about it in the literature of primary productivity. And then we're also expanding from just feeding one grazer to looking at the entire food chain and seeing how much it goes up into our predators. So with that, if there's time, I'll take questions. But remember, I started late. Stop.
So we'll take a couple questions just while we're transitioning. Apparently Rex has a question.
I don't trust him.
Wonderful talk, Jane. And just wanted a little bit of our history. You know, I taught for years at another biological station, University of Michigan's biological station, and many of my former graduate students from those classes are here. One of the things I enjoyed about teaching there, it was primarily a recruiting area for me, because bright students go to field stations in the summer, and if you could pluck them into your lab, all the better. So with Jane, I think she told me she was very bright, and she was what, majoring in
very bright.
you were majoring in English lit or something?
Yes, proud of it, proudly English literature major.
And I said, no. You need to study diatoms. So she said, well, she had to negotiate with her parents, apparently. And they said, you're leaving Michigan and going to Bowling. What are you doing? What are you going to study? And so I had to go out to dinner with her mom and dad to convince them this is legit. And Jane trained me ahead of time, and she said, "No, Rex, don't be crazy. Don't be crazy." And it all worked.
And I thank you. One more minute. Does anybody have science questions?
What happens when eutrophication during exchange? Does the Cladophora disappear? Does it get replaced? Or what stage of the eutrophication scenario is Cladophora?
Well, Cladophorabecomes is a big mystery, right? Because it is in really pristine ecosystems and really polluted ecosystems. And so there's no good answer to that. I do know that if you add nitrogen and phosphorus to Cladophora in the Eel River, the Cladophora does find the diatoms shift. So you get rid of the epithemia and then it just takes over by a different group of Knitzias and naviculas. So the diatoms are more sensitive to changes in nutrient levels of the water, at least in the eel, than the Cladophora is. Yeah.
So if you didn't get your fix on Jane, you can talk to her later.
Okay.
We're gonna transition to the next talk. We've got Gina coming up here. She's gonna continue talking about adventures in algae. Thanks to Rex.
All right.